So if you think of the human genetic code as a long document filled with letters, so what you're talking about is technology that can go into that document and you can change certain pieces of it.
That's correct. With sequencing, we've gotten good at reading the documents, and now with genome engineering and gene editing tools like CRISPR, we're getting fairly proficient at writing into that document.
So what is CRISPR?
CRISPR is a tool that allows scientists to modify the genetic code inside cells. And so you could perhaps remove a characteristic of a plant or cure a disease that's genetically based.
The DNA in the case of humans is about 4 billion bases long. So these are the Ds, Cs, Ts, and As, which specify the behavior of the cell. Now what you can do with CRISPR is you can insert an enzyme into the cell, and it comes in and it basically cuts the DNA. And so it's able to replace some of the letters with different letters. And this then can change the behavior of the cell.
As a field, CRISPR and genome engineering is very early on. We're just beginning to understand what's possible. This, as a computer engineer, is like being, say, in the 1970s with computers, where everyone had mainframes and there were very few computer users. In the future of gene therapies, you could imagine maybe 20, 30 years from now that it's possible to take a pill that ultimately completely cures a certain type of genetic disorder that's been inherited.